All right. Thank you, everyone, for taking time to attend today's talk as part of the third annual Gene and Cell Engineering Virtual Summit. So my name is Ray Miller, and I've got the privilege of introducing this session's speaker and then moderating the Q&A talk following the speaker's talk. As a reminder, please feel free to type your questions into the chat box towards the bottom of the screen. So our speaker for today is Lauren Alfonse. So Lauren is currently a principal scientist at Arbor Biotechnologies, where she's leading a team that is discovering and developing new CRISPR nucleases. Obviously a topic really relevant to the entire symposium, to the entire summit. So this project is in collaboration with a prominent external partner. So Lauren has joined Arbor from its early days and has had the honor of seeing several of their programs and assets through the journey from discovery into now preclinical relevancy. So before joining Arbor, Lauren worked at the Gergate Lab at Boston University School of Medicine. During her time there, she and a team of multidisciplinary researchers developed a new, now commercially licensed software that determines the probabilistic distribution of a number of contributing factors to complex forensic DNA profiles. So Lauren received her BS in biochemistry from Boston College and her MS in biomedical forensic science from Boston University School of Medicine. So today's talk, Lauren's talk, is titled Discovery and Engineering of a Miniature CRISPR-Cas type VL System. So Lauren, the ball is yours. Please feel free to start when you're ready.
Hi, everyone. I am Lauren Alfonse. I'm a principal scientist at Arbor Biotechnologies. I've had the great pleasure of seeing the work that I'm going to present on today from the earliest days of discovery through to more recently some preclinical work. And so I'll be talking today on our discovery and engineering of a miniature CRISPR-Cas type 5L system. And before I get started, I did want to thank GenScript for extending the invitation to present today. Okay, we're going to dive right in. So as follow-up to some of our earlier work on the discovery and development of Cas12i2, which we've both presented on and published at Arbor, we elected to explore the diversity of type 5 systems. And what we're showing on the left here is a phylogenetic tree of CRISPR-Cas type 5 systems. And we highlight the evolutionary distinct branch of type 5Ls in this yellow dot. And in the blue callout, we are showing our Arbor type 5L. So we identified a family of miniature CRISPR-Cas type 5L systems by a metagenomic search. And we later looked at this family in depth with a preliminary bacterial screen. So from that family that was highlighted on the previous slide, we did zero in on a smaller enzyme. And we put this through a screen in bacteria by taking a plasmid which encoded a library of 8,900 different spacers that target either E. coli essential genes or a PACYC plasmid, which was co-electroporated into E. coli cells. In this case, they were specifically E. clone electrocompetent cells and grown under antibiotic selection in microfluidic droplets. So the way that this screen works is that active targets interfere with bacterial growth. And so as the plasmid is cleaved, we see essentially cell death or depletion of the targets that we're examining. So on the left, we're showing the distribution of those targets on the E. coli essential genes that were targeted. And it's a very small image, but basically the orange rectangles show depletion, and they're shaded from lighter as kind of more modest depletion of that target, and a darker shade of orange on that gradient is stronger depletion. So for this system, we did see robust activity in E. coli, and we also, through the DNA sequencing that was output, determined the PAM, which was shown at right. So we do see a TTR PAM for this particular system. Given these results, we then conducted RNAseq on the bacterial cell lysate to determine the process form of the CRISPR array. So we're showing at the top the small RNA-seq reads aligned to the sequence of the CRISPR array. And so we determined that this system uses a 36 base process DR and roughly a 20 base spacer. There is a little bit of flexibility as to where you can call kind of the end of that sequence based on where the reads start to drop off. In addition, and although the data are not shown here, we did include a non-coding element in the plasmid construction where somewhere in that non-coding element may code for a tracer RNA. we determined that this system does not require a tracer RNA for nuclease activity by aligning the small RNA-seq reads back to the non-coding element that was included in the plasmid. And again, it's not shown here, but we did not see really enrichment of reads anywhere throughout the plasmid. So again, we determined that the system does not require a tracer for activity. Next, we conducted a series of biochemical experiments intended to characterize different properties of the nuclease. And one of the main ones that we were interested in is determining the cut site. And so in vitro characterization revealed that the major cut sites occur external to the spacer. So up top, we are showing where the spacer is. So that's that green line. The non-target strand is on top with the PAM at left, and the target strand is on the bottom. So the shading of the red triangles, again, is lower cut frequency is a lighter red, and the more frequently detected cuts are in the darker shade of red. So I should say that we determined these cut sites by an in vitro assay, which entailed complexing purified effector protein with the CRISPR RNA that we determined on the previous slide, and then incubating that complex with a plasmid that harbored an AAVS1 target sequence. And as I mentioned, this readout was determined by next generation sequencing of the cleavage products with the staggered cuts shown above. Given the results that we saw both in bacteria with robust activity and also with characterization of the cut site and additional biochemistry experiments that we're not showing here that sort of characterize the activity and function of our nuclease, we then put this nuclease through a screen in mammalian cells to determine if it has activity. So we're not showing it here, but we did observe low-level wild-type activity with this nuclease, which was actually fairly exciting. So we elected to then engineer the nuclease to increase that on-target activity. So the results of the engineering screen are shown at the left. And essentially what we did here was generate two libraries. So one was an arginine scanning mutagenesis library, and then the other was a glycine scanning mutagenesis library. And we screened those full libraries initially by a bacterial depletion assay. So it's very similar to what I described at the outset that actually initially resulted in the discovery of this nuclease. But for this application, we sort of tweaked the assay a bit to engineer the activity of the nuclease instead. So once we put the full libraries through the bacterial screen and we identified those residues that seemed to increase depletion activity in bacteria, we then validated those hits in mammalian cells. So that's actually what we're showing in the plot at left. We're showing indels relative to wild type. So it's just a way to normalize that signal for BioRep1 and BioRep2. And then the glycine point mutants are shown in the darker grayish black circles, and the arginine are in green. So again, at left is sort of the mammalian validation data, and the red box is highlighting those point mutants that really seem to yield the most significant increase in activity. So we were seeing levels basically of up to 2 to 4x activity relative to wild type with just a handful of point mutants, which we then proceeded to screen in combination. So we're skipping sort of the data that showed all the different combinatorial mutants that we tested, but ultimately the top arginine and glycine mutants were screened in combination and produced a combinatorial variant that we decided did show very exciting activity in mammalian cells, and that we elected to develop further. So the plot at left is showing the mammalian activity of that combinatorial variant in blue across 12 targets tested in mammalian cells. So the y-axis is showing the percent indels, and we did include an SP-Cas9 control for each target. In this case, the spacers did overlap, so it was a fairly direct comparison of the activity of SP-Cas9 to our small type 5 combinatorial variant. We were very excited to see that the activity of the combinatorial variant outperforms SP-Cas9 when you average the activity across all 12 targets tested. Given these results, so far, everything had been done by transient transfection to HEC293Ts. We next wanted to explore the translatability of our nuclease for various therapeutic applications. So we proceeded to purify the protein and develop RNP of it. And again, just emphasizing here that this is now work done on the combinatorial variant that we obtained through our engineering pipeline. And so here we tested 3 different doses of our RNP complex. So dark green is 11.5 micromolar. The middle shade of green is 5.7 micromolar, and the lightest green is 2.9 micromolar. And again, we're very excited to see robust activity, nearly saturating signal for AAVS1T1, EMX1T2, and VEGF-AT3 across all 3 doses tested, and in particular, sort of maintaining comparable or higher activity at the lowest dose tested, which was 2.9 micromolar. So given the activity observed by RNP delivery to HEX, we next decided to really try to push ourselves into preclinical work by evaluating activity at a therapeutically relevant locus in primary human hepatocytes. So we are not disclosing what locus this is, but just noting that we did test 19 different targets at this locus of interest. Again, this is by RNP delivery to primary human hepatocytes. And we did test 2 different doses. So in purple, we have 5 micromolar. So this is going to be informed by the data on the previous slide. And in light purple, we have 1.25 micromolar. We included negative controls, which were cells nucleus affected without RNP, but read out by targeted sequencing at each target site for the 19 that were tested. And these data were represent the results of 3 technical replicates. So what we're seeing is, clearly with the higher dose and also to some extent with the 1.25 micromolar dose, indels that exceed 60%, which for us was a great result and very promising for the engineering that we did on this nuclease. And finally, it's important to note that all of the work preceding this slide was done in the absence of a structure. However, we did recently determine the cryogram structure of our small type 5. And sort of in hindsight, it provides insight on the mechanism of increased activity of the engineered combinatorial mutant. So the image of the structure is shown at left. We do so this was done on the combinatorial variant as well, just a catalytically inactivated version of the ternary complex. And overall, the cryogram structure was solved at 3.2 angstroms. So we see the RevC and stop domains, as well as various regions contacting the PAM and guide RNA. So now having this structure should unlock other capabilities beyond sort of what we've done to date, which is definitely promising as we move this forward to future applications. So with that said, in summary, small size coupled with potent mammalian activity make the combinatorial engineered variant of our miniature type 5L CRISPR-Cas system an attractive candidate for therapeutic genome engineering. And lastly, I would just like to wrap up by saying thank you to members of the Arbor team, both past and present, who contributed to this work. Specifically, the authors listed below. So Alexa, Erin, and Jenna are all on my team and have done incredible work, and various others, all listed here, and also just those who have contributed throughout the years. As I mentioned at the outset, it's been a great pleasure and extremely rewarding to see this work kind of take shape over the course of my several years at Arbor. So I think with that being said, I will take any questions now.
All right, Lauren, really, really excellent talk. Thank you so much for giving that. Really exciting work that you and your team are doing. So what we're going to do now is we're going to pivot to the Q&A part of this session. So just as a reminder to the entire audience, if you have a question, and obviously we encourage questions, please type your questions into the chat box, which should be floating at the bottom of your screen. So if you need help, just give us a holler and we'll try to help you with that. So I'm going to take your questions and I'm going to read them off to you, Lauren, and appreciate your answers, your comments, your feedback. So let's go ahead, and as people are taking time to put their questions in, I'm going to exercise a little moderator's privilege here. So I'm going to ask you a question for me. So how long does it really take, your team, your company, other teams to be able to go ahead and to take a nuclease to market? What are some of the roadblocks to bringing a nuclease to market?
Yeah, I think the timeline is an interesting point. I don't think I'm sort of maybe the best poised to give an answer as to bringing it to market, but I can give at least some insights into how long this work took. So essentially what I presented on today, kind of start to finish, took about a year. In terms of how that fits into Arbor as a whole, I think it's actually interesting because being here from the start, we were founded as a company focused very broadly on biodiscovery. So the first 2 years of Arbor, we really focused on making very rapid discoveries. And then after those 2 years, it was sort of let's kind of actually develop, our clinical strategy. And so there was a little bit of a delay for this enzyme between the time at which the discovery was made and identified as an active nuclease, and then kind of when we actually picked up the enzyme, with a concerted effort to develop it, which, like I said, took about the past year.
Nice. So we've got some questions coming in from the audience now, so switch to the audience's questions. So let's see. First question. Have you considered other ways to improve the activity of your nuclease besides scanning mutagenesis?
Another great question there. So what I kind of touch on at the very end is solving the structure. So now that we have that cryogram structure of our protein, that's really unlocking the next generation of engineering for us. So sort of the scanning mutagenesis approach was a very rapid way of getting us to an engineered variant, like I said, kind of hitting the 1-year timeline that we were pursuing last year. But now that we do have the structure, there are kind of additional avenues that we're going down.
Nice. So another question here. Wonderful presentation. How do these nucleases compare to Cas12a and Cas9?
So I think sort of to orient us to Cas9 is probably the best focus. So Cas9 is a type 2. Ours is a type 5. There are 8 differences there. I think kind of thinking wise, it's much smaller than Cas9 from a protein size. The guide is quite a bit smaller as well, especially given that the guide for our enzyme doesn't require a tracer. The PAM is very different. So Cas9, out of the box has an NGG PAM. Ours is TTR. There's kind of differences in terms of cut pattern, where our nuclease cuts outside of the spacer and generates staggered cuts. In terms of indel pattern relative to Cas9, Cas9 you tend to see like +1 insertion. Our type 5, you tend to see larger deletions. So there are kind of quite a few differences between those two.
Sounds good.
I guess one last one that the data is actually quite interesting, but did not go into this talk. So ours also does self-process its own pre-cRNA, which is different than Cas9. And then also different than other type 5s that I'm aware of. This one processes in the spacer rather than in the DR.
Got it. So I think a follow-up question from a different person in the audience is, are there clear advantages for your nucleus compared to Cas9?
I think, again, sort of the philosophy of our company as a whole is sort of expanding our toolbox so that depending on, as we focus more on preclinical, as we've kind of grown as a company, having different indications that we want to go after, selecting from our toolbox what is the appropriate nuclease. So for example, if a given indication isn't accessible by an NGG PAM, you wouldn't want to use Cas9. You would want to use something that does not have an NGG PAM. So I think there are definitely advantages there, where sometimes Cas9 is an obvious choice and works really well for certain applications, and there are a lot of them. But I think sort of our approach of going after other nucleases that are smaller, theoretically easier to deliver. In this case, our nuclease, which has a smaller effector and also a smaller guide and self-processes, kind of enables all-in-one AAV delivery, which would be difficult to achieve with some of the other larger nucleases that also have larger guides.
So another question from the audience. How does one license a nuclease that Arbor has developed? Do you charge licensing fees to researchers? More of a business question. But hey.
Yeah, a little bit. Again, I'm not on the business development team, so we engage in external collaborations. I think there's some interest as a company also in partnering with academic institutions. So in terms of like the price model, though, I'm really not authorized to comment on that. So I think if there's interest, happy to connect, whoever asked the question to somebody on our business development team.
And so you'd recommend that essentially anyone that's interested should contact your business development team and they would kind of lead them through the process, I'm guessing?
Correct. Yeah. And see if there's kind of a mutual opportunity there that would make sense.
Nice. Let's see here. Another question is, what's the specificity of this nucleus?
So that also is sort of late breaking data that I think kind of the philosophy that we've gone after for this nucleus, or the model that we've used in screening at least, is take a broad pass at guides at the therapeutic locus of interest, focus on on-target activity, and then sort of follow up with a deeper, guide-seq, ramp-seq analysis for your lead guides there. So we didn't disclose it here, but in terms of just a broad answer that I can give, it is similar to what we saw with Cas12i2, which is another sort of Arbor nuclease that's a little bit more developed in our pipeline. And we have a publication on that kind of goes over the specificity in more depth. Although hoping that, in the coming months, I'll be able to also kind of disclose that with more specificity on 7763.
Nice. Do you plan disclosing that at a future conference that folks can stay attuned to, or publication?
I hope so.
Good deal. All right. Another question. You're testing the protein form of your nucleases. Do you test the mRNA form?
Yes. So in terms of what we've done typically and historically, we traditionally will do a first pass by plasma delivery to, whatever cell type, traditionally hex, then follow that up with I would say like a more thorough characterization by RNP delivery to, again, a different cell type, maybe the same as in the first pass or not. And then sort of the third strategy is mRNA delivery. So I didn't show any mRNA delivery data here, but that is sort of part of our workflow as well.
Nice. I guess I'll ask my follow-up question to the audience members' question here. Do you deliver mRNA through lipofectamine, or do you use electroporation like you're already doing for RNPs, or?
Yeah, kind of a couple of different methods. It depends on the application. So.
Let's see here. I don't think we have any more questions from the audience here. So I think unless there's any further questions, Lauren, really want to thank you for your time. Excellent talk, and thank you for staying and answering questions from the entire audience. It's really greatly appreciated. I also want to thank the entire audience for taking time out of your schedule to attend this session, and then also to attend the broader Gene and Cell Engineering Summit. So we'll go ahead and we'll close the session. Thank you all so much, and have a great day, and goodbye.
Thank you.
Thank you.